So this is the liquid broth medium where you can see only the liquid culture. This first picture is the nutrient broth before inoculation of the E. coli, and after inoculating that nutrient broth with E. coli you could able to see the difference. So first test tube is very clear solution and transparent solution where the after incubation of 24 hours at 37 degrees Celsius you could see some of the turbidity maintained in that test tube. So this is all about the liquid medium. So coming to the types of culture based on chemical composition. So apart from the agar we have adding, we want to add some of the enriched products for the efficacy of the culture medium for the growth of microorganism. And there are several types like basal media, enriched media, selective media, enrichment media, indicator media or differential media, transport media and storage media. So coming to the basal media, it enhances the growth of many microorganisms and mainly it contains carbon and nitrogen, and it is used in laboratory for subculturing the microorganism, and it is non-selective media. It is for all microorganisms, not a selective purpose, and it supports the growth of mainly Staphylococcus aureus and Enterobacteriaceae species, and examples are nutrient agar, peptone water. And coming to the enriched media, so the enriched term in the sense it has to be enriched with some of the additional substances like blood, egg or serum. So in normal agar you want to add blood, so that blood agar is the enriched media for the microorganism, and examples are blood agar and chocolate agar. Blood agar mainly identifies the hemolytic bacteria. So here hemolytic in the sense the bacteria can able to lyse the red blood cells, so disrupt the red blood cells, that is the term used, hemolytic. And chocolate agar it mainly supports the growth of Neisseria gonorrhoeae species. And selective medium, so it promotes the growth of specific microorganism. For if suppose nutrient agar is common for all microorganisms to grow, but only the selective media will able to grow specific microorganisms. For example, Salmonella shigella agar. So this specific Salmonella shigella agar mainly isolate the Salmonella species. So if you are caused by the typhoid fever or paratyphoid fever, the Salmonella organism is responsible for causing that disease. So for that identifying Salmonella, we will be using Salmonella shigella agar. So this is a very good example. And coming to the differential media. So differential media shows visible changes due to an indicator differentiating the bacteria. So gram positive bacteria and gram negative bacteria, this is mainly the differential media. So for gram positive there may be a different media, and gram negative there may be a different media. So function, it distinguishes bacteria by colony color. So while gram positive bacteria exhibit in purple colonies, while gram negative bacteria appear in red color colonies. So to differentiate the colony by color, this differential media is helpful, and the indicators which will be used for this media is neutral red indicator, phenol red indicator and methylene blue indicator. For example, mannitol salt agar and blood agar. And this is the differential media where you can differentiate the species. So in that second you could see the Staphylococcus aureus and Serratia species on specific agar. It should appear as red yellow zone, and this appear as orange zone. So this is the example of differential. And types of culture based on the oxygen requirement. So based on the oxygen requirement, there are divided into two types: aerobic media and anaerobic media. Aerobic in the sense the presence of air, the microorganism can able to grow, and in the absence of air, the microorganism can able to grow as anaerobic media. So aerobic media supports the growth of microorganism requiring oxygen, and cultivating mainly easy on solid media, and growth suitable for non-fastidious microorganism. Example, peptone water, nutrient agar. So peptone water is mainly for maintaining the carbon and nitrogen. So this is aerobic. And in anaerobic, it cultivates the anaerobic bacteria by maintaining no oxygen or very low level of oxygen. And preparation, boil the water bath to remove dissolved oxygen. So the oxygen should be dissolved so that no oxygen will be taken place. Example, RCM, Robertson cooked medium. So this is the Robertson cooked meat for anaerobic media. And coming to the applications, this culture media mainly used for culturing the microbes, isolating pure cultures and identifying their characteristics. And this is also used to test for microbial contamination samples of different diseases. And it mainly to observe the colony type, colony color, colony shape and colony cause, and mainly used for the antibiotic sensitivity test.